Hi everybody and welcome to our today's second online symposium from Abberior. My name is Julia Menzel. I'm an application specialist at Abberior and I am your host for today's session. And today we have a really cool topic. Today we talk about nanoscopy in plant cells, which can be a really difficult sample. And therefore I'm happy to introduce to you our today's experts. We have here Kathryn Wright, who is a senior scientist and microscopist from the James Hutton Institute, and she has more than 30 years of experience in plant cell microscopy, which she will share with us today. And as a second speaker, I'm happy to introduce my colleague Jan-Gero Schloetel, who is an expert for STED microscopy with a preference for tricky samples. And Jan-Gero will talk about nanoscopy in plant cells and give a short live presentation with our facility line and tobacco plant cells. And before I hand over the word to our speakers, I would like to remind you to use the chat function to ask your questions during the talk. And with this, I hand over the word to Kathryn Wright. We are looking forward to your talk.
Thank you for the introduction and for the opportunity to describe some of our imaging work on plants and plant pathogens. I'll leave my camera switched off so I don't get in the way of the slides. I work at the James Hutton Institute in Dundee. We undertake research for customers, including the Scottish and UK governments, focusing on some of the major crops important to the Scottish economy, including potato, barley, and soft fruit, including raspberries and blackcurrants. As far as imaging is concerned, much of our work is related to the investigations of plant pathogens, including Phytophthora in potato, the fungal pathogens Rhynchosporium and Ramularia in barley, and more recently for soft fruit, the raspberry root rot. I started working with microscopes over 30 years ago with Carla Parker, using fluorescent probes to look at dye uptake and solute transport. Since then I've worked on various projects and plant tissues, including, as we can see here, oat alleurone protoplasts, Arabidopsis roots to study symplastic unloading pathways, and plant viruses including tobacco mosaic virus, potato mop-top virus, and potato virus X, which forms these rather intricate structures as viral replication factories. Here showing actin in magenta and the viral coat protein in green. Most of the imaging we do involves live samples with minimum tissue preparation. We do this to try and avoid disturbing the plant pathogen interactions. For the plant cell imaging, we work with upright microscopes fitted with water dipping lenses, and these often allow us to image in direct contact with the tissue. We've found it's difficult to mount leaf tissues securely under a cover slip to work on inverted systems and to image as deep into the tissue as we need. We've progressed through a series of confocal microscopes as detection sensitivity and resolution have improved, and we're looking forward to being able to use super resolution imaging in our investigations. However, as I hope you will see in the next few slides, this might prove to be challenging. First of all, let me introduce you to some of the work we've done on barley, an important crop to Scotland and its whisky industry. Barley is challenging to image due to a number of features. We can see here on the left, in common with many plant tissues, using a confocal with sensitive detectors we can easily detect autofluorescence in all the common excitation-emission combinations. At times this can be useful. Here it has allowed us to detect the distribution pattern of the stomata in mutant strains of barley. But at other times it causes interference. Chlorophyll in particular can cause severe issues. Barley leaves have a very hydrophobic cuticle, which means for higher resolution imaging they repel the water required for our dipping lenses. These leaf segments have therefore been mounted in silicon oil under a cover slip held in place by double-sided tape. This hydrophobicity also means it's difficult to stain tissues with the usual fluorescent probes without adding a surfactant. Here I've used Silwet to allow the tissues to wet. Therefore, there is a potential to disrupt the plant cells. Even if the tissue is stained, imaging is challenging. These epidermal cells only contain a very thin layer of cytoplasm. And as we can see in this XZ section, without depth compensation, the fluorescence signal is rapidly quenched in the underlying mesophyll cells. You will also notice that this tissue is not flat, meaning we were looking at very deep Z sections. The reason I started imaging barley was to investigate the infection process of this fungal pathogen, Vincosporium. This causes a disease called leaf blotch or scald and can lead to a reduction in yield of up to 40%. I stained both the barley and fungal cells with carboxymethylfluorescein diacetate. This accumulates in the plant cell vacuoles so that we can see when the plant cells are still intact. Also, staining with pyridium iodide allows us to see the plant cell walls, and then it will stain the nuclei when the plant cells start to collapse. My staining method has been largely replaced after one of our PhD students, Amar, successfully transformed Vincosporium to express green fluorescent protein. We can't see when the plant cells start to collapse until the chlorophyll in the mesophyll cells becomes disrupted. However, using this transformant allows us to identify cultivars showing resistance to the fungus. We can see a more random distribution pattern on resistant strains. And this helps us to identify genes involved in this resistance, and this feeds into our barley breeding program. Amar also transformed another barley pathogen, which causes ramularia leaf spot. This forms thin hyphae running on the surface of the epidermal cells until they penetrate a stomatal pore. They then form larger diameter mycelia, filling the substomatal cavity, before producing reproductive characteristic swan-necked conidiophores. It would be interesting if we could investigate these interactions at higher resolution. Moving on to the potato pathogens, we often use Nicotiana benthamiana, a tobacco species, as an alternative host, or for agrobacterium-mediated transient expression of pathogen proteins. We have a collection of stable transgenic plants expressing fluorescent proteins targeted to various cell organelles for co-localization studies. For those of you expecting to see the rectangular boxes of onion epidermal cells, here we're looking at the jigsaw-shaped cells of benthamiana epidermal cells, with the paired lips of the guard cells surrounding the stomatal pore. Since these are all pseudo-coloured images, green fluorescent protein is usually represented in green, RFP in magenta, leaving the autofluorescence from chloroplasts in blue. In these pictures we can see the network of the endoplasmic reticulum, which we will see again later in the demonstration, with long transvacuolar strands and a membrane continuous with the nuclear membrane. When we investigate cell-to-cell transport, we often look at plasmodesmata. These are the pores connecting the adjacent cells. I know a number of my virologist colleagues are very interested in looking at these with super resolution microscopy. Here are a couple of pictures illustrating the leaf structure of Nicotiana benthamiana. To enhance depth penetration, this leaf has been mounted in perfluorodecalim. Unfortunately, since this is water immiscible, its use is not compatible with imaging plant pathogens which cause cell disruption. If we look at the upper leaf surface, there is a layer of epidermal cells above the closely packed palisade mesophyll cells. Above the epidermis is a waxy cuticle, and this can cause reflection on autofluorescence problems, and it is part of the reason we usually image the abaxial leaf surface. Again, here there is a layer of epidermal cells containing a few chloroplasts above the spongy mesophyll cells with numerous chloroplasts. Between the cells are large air spaces, allowing infiltration of, in this case, perfluorodecalim, but usually either water or fluorescent probes. This prevents air escaping through the stomata during imaging. With sensitive detectors and depth compensation methods, it may be possible to image below the mesophyll cells to the vascular bundles. One of the major potato pathogens, studied by a group from Dundee University based at the Institute, is Phytophthora infestans, the cause of late blight. These pictures have been taken by Petra Bovink and her colleagues in Paul Birch's group, and show that many of the interactions taking place between the plants and this uromycete pathogen occur deep in the leaf tissue. They are particularly interested in the proteins expressed and secreted by Hostoria, including cytoplasmic and apoplastic effectors and cell wall degrading enzymes, probably involved in suppressing plant defenses during the biotrophic growth phase. Here and in the background, we can see an effector protein which is specifically secreted at Hostoria, and a more detailed image of effectors in magenta around these Hostoria seen in a 3D projection. Just to emphasize the depth of imaging required, here we have a depth-coded image with the aposoria on the surface of the epidermis, down at least 50 microns into the tissue down to the Hostoria. It is hoped that super-resolution imaging will help resolve the detail around these Hostoria, including in which membranes proteins are expressed, whether they are secreted, and any vesicular activity. Moving away from plant pathogens, here we have the human pathogen Escherichia coli O157H7. There have been numerous outbreaks of food poisoning in recent years caused by E. coli on sprouted seeds and associated with romaine lettuce, and it is now clear that E. coli O157 can use plants as an alternative host. I infiltrated Nicotiana benthamiana leaves with the bacteria expressing green fluorescent protein, and you can see that they form large colonies here inside the leaf underneath the epidermal cells, and also deeper near the vascular bundles. From the food point of view, the good news is that not all plants are as susceptible as Nicotiana benthamiana to the bacteria. Here in spinach or tomato, I've inoculated with bacteria expressing both green and red fluorescent protein, and these show that only a few cell divisions have occurred. By infiltrating stain into the leaf, we can see that the colonies are showing characteristics of biofilm formation, producing curly fibres which are associated with cell adhesion, here stained with Congo red. Further evidence was provided by the expression of this transcriptional reporter fusion to the structural components of curly. As I mentioned previously, E. coli has been associated with many outbreaks of food poisoning involving sprouted seeds. Here we can see that a similar system involving young germinating seedlings can also be extensively colonized. These are microgreens, often used as nutritional and attractive garnishes. For example, broccoli, amaranth and basil. And as you will be able to see, as well as form extensive colonies on the surface of the cotyledons, occasionally the bacteria can enter through the stomata and form internal colonies. Again, these bacteria are forming biofilms on the surface of the cotyledons. They're often located in the channels of the anticlinal walls, possibly due to the increased water supply or protection provided in these channels, but also associated with the supply of arabinose, which the bacteria can transport and metabolize. Therefore, these young germinating seedlings can act as a host for E. coli, and the formation of these biofilms means that bacteria are more resistant to removal by washing. This means that, as with sprouted seeds, the production should be controlled to prevent the use of contaminated water or contaminated seed stocks. In summary, I hope I have been able to show you some of the challenges faced whilst imaging pathogens in plant systems, caused by autofluorescence, the presence of the cell wall, and the depth of tissue we need to study. To move to super resolution imaging of these systems, we predict that the features of spectral detection and time gating will help deal with the fluorescence from chlorophyll. Adaptive illumination will help minimize the laser power required, and adaptive optics will help us penetrate deeper into the plant tissues. Finally, I'd like to thank my colleagues involved in some of this work. Alison, who leads the Imaging Technologies group, as well as studying virus and solute transport. Thanks to Amar, who transformed the Rhynchosporium and Rammularia and provided some of the barley images. To Petra and Dion for the Phytophthora images. I acknowledge the Scottish Government and our various grant awarding bodies for funding this work, and thank you for your attention.
Thank you, Kathryn, for your nice talk and for sharing with us your tips and tricks about plant microscopy. And the Q&A session we will do later as a shared session between you and Gero. And now I will hand over the word to our next speaker, Gero. Gero, the stage is yours.
Thank you, Julia. Let's get started. Before we come to the interesting part, imaging on our facility line microscope here, I would like to give you a quick introduction. I would like to talk about the technologies we can offer on our facility line platform, and that ranges from confocal microscopy to super resolution microscopy. In confocal microscopy, we can offer a few advanced detection schemes which can improve imaging for plant scientists. We also offer matrix detection on our facility line microscope that improves the signal, reduces the background, and can improve resolution. It's very easy to use, and it's also combined with live confocal or STED microscopy. When we're coming to STED microscopy, this is the highest resolution microscopy on this machine, and we can get below 30 nanometer resolution. And in the Z-axis, so vertically, we can go below 75 nanometer resolution. It's a standard method, it's our bread and butter method, and it is also live cell compatible, and we have a few tricks to make it even more live cell compatible. On the other platforms, we have a few alternative methods, for example, resolved microscopy, which is using specialized fluorescent proteins and can reach about 50 nanometer resolution in a really good sample. And also on a different platform, we have MinFlux microscopy, and in MinFlux microscopy, we can reach below 2 nanometer resolution, meaning really molecular resolution. This is our microscope, the facility line, and when we look at its confocal features, we can see that it's a full-blown confocal microscope. We have lots of excitation lasers to cover all the dyes we would like to look at. Beam scanner, obviously. We have a motorized pinhole, which we can adapt in size. Spectral detection, which we come to in a minute. We have a wide range of objectives. Time gating for the detection. Transmitted and reflected light, which is important for plant microscopy if you want to get some orientation in the sample. And a very intuitive software platform. The STED features of this microscope involve two pulse STED lasers, 595 and 775 nanometers, to cover all dyes from green to red with super resolution. The resolution, as I mentioned, 30 nanometers in XY or better, and 75 nanometers in Z. We have auto alignment, which means you have one push of a button to align the whole system. Easy 3D STED is doing the 3D super resolution on our system. And we have adaptive illumination to reduce the light dose going into our sample, which means we get less bleaching, less phototoxicity, and better resolution. We also have a continuous autofocus, which works in confocal and STED mode. We have adaptive optics to go deeper into our samples. And if you like, film upgrades and further upgrades are possible even on an older microscope here, you don't always have to buy the newest model to get the newest tech from us. Coming to the confocal features here. Well, if you're looking at confocal microscopy, we have to think about the natural dyes present in plants. And maybe the most dominant ones, or which we would always think of, are chlorophyll A and B, and carotene, and so on. Chlorophyll A and B absorb here in the range where you would excite blue and green dyes, but also absorb again in the range of 650 to 700 nanometers. And where would the fluorescence be once we've excited these dyes? Well, there's a whole bunch of other natural plant dyes as well, but looking at chlorophyll, that will mainly fluoresce in the very red range, and the carotene would, for example, fluoresce more around the green range here. However, chlorophyll is really the dominant or strongest part, at least in the samples we are looking at today. So what can we do to suppress autofluorescence and get to the real signal? Well, first of all, we have spectral detection, which means we can narrow the detection window for each detector to a certain wavelength range, which avoids then most of the autofluorescence. So that's a good thing, and it's very easy to use in our software, as I will show you in a minute. This works with 4 detectors at the same time as well, each of them selecting a spectral range. The detectors then have a APD, so a avalanche photodiode. We are using these because they're especially efficient at detecting light. That's important, as Kat mentioned, to go deeper into your samples, for example, and also to get a very bright STED image. And our APDs are superior to most detectors in detection efficiency in the red range, and in the green range they're at least on par, if not better as well. These detectors are also photon counting, which means we can have a detection window with a delay. So what does that mean? This means we get time-gated detection by using our pulsed excitation lasers. And if we look here at the time axis, the excitation pulse is very short in time. The fluorescence, on the other hand, is stretched out over time, most intense directly after the excitation pulse, but decays over time. And when we say time-gated detection, that really means we have a detection window here, which has a certain delay after the excitation pulse, and then a certain length for detection. What's the advantage here? Well, if you imagine the autofluorescence decays much quicker in many cases than the intended fluorescence of, say, fluorescent proteins or stains, that means we can wait until this autofluorescence has decayed and then look at the later photons which are free of autofluorescence. Why do we have time-gated detection? Well, that's because we are using a completely pulsed system with also a pulsed STED laser. And that allows us really to wait until the STED pulse has done its job, meaning giving us super resolution, and then only detect photons coming after this job has been completed, meaning we're getting the sharpest image for the least irradiation of the sample. I've invaded the leaves with water before putting them on the microscope. And now I'm going to use the autofocus to find our focal plane or the cover slip. Okay, and now I'm switching to the software. And here we have a choice of dyes. I can also select my dyes according to the wavelengths where they emit. So that's very handy. Or just go to the recent dyes. And there I have EGFP for our EGFP tubulin. And we also have autofluorescence. Here, the chloroplast. How did I make these? So here you see a customization menu, which I can do for any dye. And I can adjust the spectral range of absorption and fluorescence here and set up basically a new autofluorescent dye. I can also do this for carotene or any other dye. Now I'm just naming our sample here. And I'll start a new overview. I can select how large my overview should be when I'm using the tiling mode. I can also select if I want overlaps, which is important for stitching. just go to live imaging now. And when I select live imaging, I will get a large overview. Basically, the whole field of view you can record with a scanner. What we can see here on the left is the chloroplast and on the right the GFP. But I can also see it's a little bit weak, the GFP. So I'm just cranking up the excitation laser a little bit higher. Now we have a nice and bright tubulin image. And I can drag the stage position here with my mouse. So you see this hand symbol, and I can just drag this, and now I'm moving the stoma here back into the center of our field of view. I can also use my mouse wheel to focus. You can see now I'm moving the focal position here. So I can navigate just using my mouse. I don't really need to use the joystick or focus wheel on the microscope. There's also a small indicator in the center of the screen, which is our Z slider, and you can see it moving whenever I focus. Once I'm happy, I stop my overview scan, and here I can switch on transmitted light. We'll also have to adjust the gain of this detector. And now in live mode, we can see again chloroplast fluorescence, tubulin fluorescence, and transmitted light. I can overlay everything nicely using these dye chips and adapt the brightness as well of each image, or I can hide images as well if they're disturbing me, but if I want to use them later again. I'll just stop this overview here. I deactivate transmitted light, and I will go to the light path menu, and here we'll adjust the pinhole a little bit, because for the green I would like to have a slightly smaller pinhole than for the red dyes. Adjust also the dwell time to make the overview a bit faster. And now if I scan and leave the scan running, I will go into tiling mode, meaning I'm moving the stage here in a spiral pattern to explore basically from our original position, which was this stoma in the center. And now I'm moving, as I say, in a spiral pattern around with the stage to explore the area. And that can be really helpful to get a nice large overview and then pick areas which might be biologically relevant or interesting. And we can nicely zoom into these areas later on. So if you, for example, want to have a coffee break, you could leave this running for a long time and then pick and choose your interesting areas from a large overview. And I will stop this scan a little bit earlier than the defined large range. So I can stop this at any point, just for time reasons here. There we go. Now I can select a region of interest, just drawing this with a mouse. I can also tilt that region of interest if I have diagonal structures. I can make it smaller and larger. And you see indicated here the exact size of the region of interest as well. And if I press live, I will automatically zoom into this ROI, or region of interest, and have a finer pixelated image of my area here. Now, if I expand this menu, I can make the pixel size larger or smaller. And then scan again. And you can also adjust the dwell time here. Scanning now. That's a little bit faster. And I can see here my chloroplasts. And if I look into my GFP image, I can see a slight shadow here of the chloroplast fluorescence. I will make the dwell time a bit longer to get a little bit more signal, and then we'll see if we can really see some chloroplast fluorescence. Yeah, I think there's a very slight green fluorescence here, which is caused by chloroplast or chlorophyll fluorescence. Now, if I go to light path and expand that menu, I can now adjust our spectral range for the two detectors here. I'm moving the GFP fluorescence to a longer wavelength range, meaning our GFP fluorescence will be much weaker, but I will also now catch a lot more autofluorescence from chlorophyll. And here we can see really, really strongly our chloroplasts now, and they are in the GFP image where they shouldn't be. If I go to time gating here, I can now activate time gating, which I described earlier for the GFP channel. And let's see what the effect is of that, just recording other versus with the same settings. And we can see, despite these really bad spectral settings which I chose, the chloroplasts now disappear completely from the image. So switching between the images really makes it clear. And here in the overlay, we can see originally we had lots of crosstalk, and switching here between them, you can see time gating really gets rid of the crosstalk, even when we're using bad settings spectral. Going back to the original image here, I can record this again without time gating, but with the correct spectral settings. So this is the default settings, and there's only very little crosstalk. If we now switch on time gating and repeat this, we can see that even the last bit of autofluorescence here from the chloroplast has disappeared, and we really have just black holes where we would have slight crosstalk with the non-gated settings. So really good tool here to get rid of the chloroplast autofluorescence. We have a new detection mode, which is using our matrix detector. That's not one detector, but actually a whole bunch of detectors. A little bit like a compound eye from insects. And each of these detectors is an APD, meaning it's a very highly efficient detector. And each of these detectors has its own point of view. That in turn means that each detector sees the detected light from a different angle. And this way we can actually discriminate between the light that comes right from the focal volume and the light which is out of focus. So we can discriminate out of focus light and wanted or intended signal. That will improve signal to noise, and it can also improve our Z-sectioning capabilities and resolution in general. We'll return to our sample, which is still on the microscope, the tobacco leaf with tubulin GFP. Here we are back in the software, and we can see the image which we have just recorded. I would like to pick one of the images where we're using the correct spectral range for GFP. And then I go to the tree view and I drag and drop this into the overview. That duplicates the image, and we can start fresh with a new image, but with the correct settings which we have already established. I'll go to the dwell time here and adjust this up so we can collect a little bit more signal. I will also deactivate the autofluorescence or chloroplast channel. And when I then scan, I will get just one green image. And here we can see several bright microtubules, but also in the stoma quite a few densely packed structures. What we can see is that there's also a little bit of haze from out of focus structures in the densely packed areas. I'll crank up the excitation a little bit more, record this once more, just to get a little bit more signal. And again, quite a lot of densely packed structures here. What I would like to do now is record this again with matrix detection. How do I do this? Well, there's really just one slider we need, and that's the matrix slider here. Now it's activated, but I will also go to light path to open the pinhole. I'll open this really wide. In a normal microscope, this would give you a very bright but also very blurred image. So why am I doing this? Well, basically, because I'm using the matrix detector, I can open the pinhole and detect as much light as possible. However, I will then get an image which is just as sharp as an image recorded with a very small pinhole. And this is possible because the matrix detector can decide what of all this light is in focus and what is out of focus. Basically, we're not wasting fluorescence, but we are also increasing the resolution. Here you can see already an image when we are acquiring the matrix image. What is this? This is a virtual pinhole image. So basically, we're calculating from all the detectors here a an image as if we're using a pinhole of different sizes. And this is done immediately while we're recording, and you can see it looks very similar to the normal confocal image. But we can do more with a matrix detector. So let's go to post-processing, differential detection. This is important. It's also calculated very quickly, but you can see the difference. Now we're rejecting lots of autofocus light, and we can see very fine structures in the densely packed areas. So what is previously a little bit hidden in haze becomes visible. The fine structures become more sharp. If I use a little bit less of this post-processing, so less sharp, I will get also a nice image, but there's also a sharpening function or deconvolution function. See here? And that will do a slight deconvolution to increase the resolution a little bit more. Comparing this to the confocal image with an adjusted lookup table, we can see we can't really get rid of the haze just by using a different lookup table. We need the matrix post-processing to get a really sharp image with all the autofocus light rejected. You can also see there's still a little bit of chloroplast autofluorescence there, but also we can see in the sharp structures here, the density areas, you can see the difference really. Now I would like to change the sample for the next part of the demonstration. And we're going for a sample or a leaf expressing a tag RFP protein. Using the autofocus here, we can now return to the presentation. Here's a movie, basically rendering of a volume acquired in confocal and matrix mode. So on the left we see the normal confocal image, on the right the matrix in differential detection mode. You can already see gaps which are not visible here in the confocal mode, and then with deconvolution on top you can see a lot more detail and gaps which are usually obscured by haze. With this I would like to finish the matrix part and move on to STED microscopy. STED microscopy will be familiar to many of you. It's a confocal scanning method, and the normal confocal image, these particles here on the left, which are really 20 nanometers in size, appear much larger, and this is because our confocal scanning spot is a size of about 200 to 220 nanometers, depending on the wavelength. Therefore, the cross section of each bead will be around 220 nanometers too. If we now add a STED laser and focus this in a donut shape around the normal laser scanning spot, we can use the STED laser to switch off the fluorophores, the excited fluorophores in the periphery of the scanning spot. And that effectively makes the PSF or scanning spot smaller. And you can see the beads now appear to be around 130 nanometer in size. What happens if we increase the STED laser power? Well, basically only in the very center of this donut, the fluorescence is allowed to persist. In all the other red areas here, we are switching any excited fluorophore completely off. So effectively, the scanning spot becomes very small here in the center, and the beads appear nearly their original 20 nanometer size. So we can gain a lot of resolution by adding a STED laser in a donut shape and increasing the intensity of this STED laser. This principle has been used recently in a few publications in fixed plant nuclei, and here the interest was mainly in meiosis. This is an example in PNAS from Raphael Mercier, one of our customers. And they were looking at zip proteins. In particular, there are N and C terminus here. And you can see that the N termini are very close together. And you see in the STED image really just one green line here for the N termini. For the C termini, which are further apart, you can actually see the two lines which you would not be able to see in a normal confocal image. Another paper from Marie-Therese Kurzbauer and Peter Schloetel and Jason Sims. They were also looking at zip proteins, but also other DNA-associated proteins, which give you more clustered patterns here. I myself had a look at samples from Isabelle Colas, and here we can see tinsel structures in a confocal image. And when we switch to STED, we can see actually not only here's a loop, but also that we have two larger bands here. And if you look closely, you can see that each of these bands is actually consisting of two very small bands as well. Again, it's not entirely visible because the labeling is not completely in every single spot, but yeah, there's two little bands here, you can see that. Now, what becomes interesting when we're going to live plant tissue is again the natural plant dyes. Here we are seeing again chlorophyll and carotene, and what is important is our STED lasers, the 595 nanometer STED laser for green fluorophores and the STED 775 nanometer for red and orange fluorophores may interfere with the plant dyes. So the 595 laser will basically excite plant dyes a little bit, and since this is a powerful laser, we might have some interference here with the chloroplasts, for example. On the other hand, the 775 nanometer laser is acting in an area where there's basically no natural absorption here, which means we can really use this as much as we want without any toxicity or noticeable effect on the chloroplasts. And with this, I would like to go to the microscope again, and we're looking at a tobacco leaf with ER tagged with tag RFP. Here we are back in the software. I'm going to start with a fresh sample. I'm selecting tag RFP and chloroplast, and I'm giving this a name, tag RFP ER. So that's an endoplasmatic reticulum. Also select a few different colors here. And I'll start directly with a live image. You can see here the ER structures and the stoma. I can now again use my mouse to move the stage, move everything into the center. And when I'm happy, I can stop this, and I will open the light path and change the spectrum slightly here, just to collect a little bit more of the fluorescence. Close the pinhole a little bit and make the dwell time a little bit shorter, and then we can scan a little bit faster. There we go. In an overlay, now I get these colors which I have pre-selected at the start. And I can focus a little bit through this stoma here. And what I would like to do is, I would like to use STED imaging in a part where I have also chloroplasts, just to show that the chloroplasts won't be harmed by the STED laser here. Let's stop here, select the region of interest, just to zoom into this area a little bit more. Make the pixels a little bit larger. And there we go. And you can see the ER structures are quite complex here in the stoma, with gaps where the chloroplasts are sitting. I will now select an area where there's chloroplasts and ER structures. And then I will change the settings for STED imaging. Here you can see chloroplasts, bring them into focus a little bit more. And now I'll switch on STED, get a little bit more excitation power, and maybe 15% STED laser power. Pixels need to be smaller because now we're looking at structures with a better resolution. And then we can start with that scan here. At 15% STED laser power, we see only a marginal increase in resolution here, but we can see a little bit more finer structures on the bottom here. I'll crank the STED laser up to 30%, and we can now see more detail on the top right corner here. And what we can also see is that the chloroplast didn't change shape or disappear or anything. Here we can see on the top right again the structure. I'll duplicate the chloroplast image. Now we have two identical images here. On the right, confocal, on the left, STED. I will go back to the overview and use a line roy here. What does line roy tool do? We can now do a vertical slice through this structure. If I go to live mode, you can see now a vertical slice through the stoma. I'll adjust the Z range here with S2, and we can see now the image going further down. We see a few structures there as well. I'll ramp up the excitation power a little bit just to see a little bit more on the bottom here. And thanks to our IPDs, we can really see structures also deeper into the leaf. Go a little bit further. There we go. I will now go to our STED image. And from there, I can transfer the settings to my vertical image. Now we have already the STED on. I just need to use this slider here to go from 2D to 3D STED. And I make the size of the pixels larger in XY because we have less resolution there, but much smaller in Z because we have a lot more resolution in Z when we use 3D STED. You can see this now in the scan. I'm going to overlay the images again, duplicate the autofluorescence channel, and we can see on the left the 3D STED image, on the right the normal confocal image. And we can see a clearer image on the left with 3D STED, more details there. But we'll also see if this is working further down in the sample. So there we can also see sharper structures here on the bottom, deeper into the leaf with 3D STED compared to the normal confocal image on the right. I can also use here a zoom tool to look at these things in more detail there. I can zoom in a little bit and also in the confocal image. And we can see that things which appear here as one large blob will be multiple structures in the STED image. I can also adjust the lookup table just a little bit here. But now let's zoom into the structures on the bottom. And on the bottom, we have also nice resolution here in 3D STED mode. And on the right, we can see the confocal equivalent being blurred much more. And now adjusting the brightness back again to the original level. And this was just to show you that we can use STED 775 nanometers without harming the chloroplasts, and also to gain resolution in 3D. This is a good point where we can switch the sample again for the following part. And again, autofocus. And we are returning to the presentation. As you've noticed in the last image we recorded, we were getting super resolution in Z and X and Y. So this is called 3D STED. And what is this exactly? Well, if we look here at a confocal scanning spot, it's about 200 nanometer size in X and Y. And in the Z-axis, it's about 600 nanometers long. When we're using normal 2D STED, we will get a much better resolution in the XY axis. But along the Z-axis, the scanning spot is still just as long as a confocal scanning spot. But with 3D STED, we get better resolution in all 3 dimensions. So we're sacrificing a little bit of XY resolution, but we get a much improved resolution in Z. And this is what we were using in the live plant tissue here. On the other hand, I've used this also in fixed samples, again from Isabelle Colas. And here we can see a nuclei here. And it's exactly the same structure as before with the tinsels. But we can see now this resolved in 3D super resolution. Now I'd like to come to a different topic, and that's adaptive illumination. Adaptive illumination is our key tool to reduce phototoxicity and photobleaching in STED microscopy. Let's go back to the presentation. And here we can see a typical sample. Typical because, well, not all the sample is filled with fluorescent structures. There are some point-like or sharp structures in the sample. And when we are doing conventional STED microscopy, we will switch on the STED laser to see these structures in super resolution. And the STED laser is constantly on, and we're scanning the whole image area or sample area with it. Now, adaptive illumination will basically block most of this STED light, and the sample is not exposed to it. However, where the STED laser is needed to give you super resolution, it is led through and will illuminate only these spots where it's needed to create the super resolution effect. And that really reduces the light dose dramatically. And I will show you this now in a live sample. And again, it's a tobacco leaf, this time with endoplasmatic reticulum tagged with GFP. Here we are back in the software. I will create a new sample with eGFP and chloroplasts again. Name this ER eGFP and go to a new overview and clean up the menus here a little bit and close the pinhole a bit further. And then I go to the slider here to get a little bit more speed, and then we will create a live overview. Here we go. And overlaying both, we can see here a stoma with lots of chloroplasts, and then areas around this with fewer chloroplasts. And I can use my mouse wheel again to refocus here. We're going to look for a nice area with nice ER structures. And I think there's an interesting area. Let's stop the scan here and select this area here. In live mode, we can zoom in here and we see a few string-like structures and a few more sheet-like structures. Zooming in a little bit further, I can adjust my region of interest to exactly the size I would like. So here about 5 × 10 micron maybe. And there. If I scan there, yeah, we're now in the right area. I will activate STED for the GFP, and I will change the dwell time a little bit. Accumulations are also changed manually here a little bit. Crank up the excitation slightly. And we also need to go to adaptive illumination. And we activate adaptive illumination here for eGFP. And this is the level above which we will use the STED laser. So in the black areas, we have low counts, and we will not activate the STED laser there. Pixel size, adapt this to about 35 nanometers here. And we will also deactivate the chloroplasts because there's none here. Now, if I scan here, we will get a confocal and also STED images. And we also see an overlay here. I will now activate the timer basically doing 50 frames with 2 seconds per frame. Pressing scan, we can see the image being recorded. I think it's moving slightly up here, so I'll refocus a little bit. Yeah, that was the wrong direction. There we go. And this is what Kat also mentioned, that the plant sometimes can move a little bit. And now we can see our structures here on the top left in confocal mode and more detail on the right and on the bottom. On the bottom, the overlay shows you in gray where exactly we are switching off the STED laser. So in the gray areas on the bottom, we are not using the STED laser. So in these pixels, no illumination with a STED light. And that's interesting because that will also protect the neighboring regions, because the STED laser is much bigger than 1 pixel. So each pixel where you switch it off also protects the neighboring bright areas. And as we set the level here to 1, every pixel which is brighter than 1 count in the confocal image is being illuminated in super resolution mode. We can see this everywhere where we have a structure, we get a proper nice STED image here. But in total, the light dose is reduced a lot. I think here in the center of the image, we can also see that this is slightly 3-dimensional, this part of the cell. So on the sides, we have more structures in focus. And if I change the focus a little bit, we can see the center a little bit brighter again. But here we were recording 50 frames easily without any bleaching. And it was a very nice movie. Blowing this up a little bit, we look a little bit at the details here, using maybe the scrolling tool here for movies, time, and let's have a look to a nice frame. I think here we can see a nice little circle at the end of this structure. If we go into the confocal image, we can see that we cannot identify this as a hollow structure there. And I think the scan speed here is sufficient to depict these really, really highly dynamic structures as well. Okay, I believe there's one last thing left to show, and that's the combination of matrix detection and STED. Since I promised this at the start of the presentation, let's try to do this in the sample. I'm going to switch to the software. Here we create a new sample, eGFP, name this is ER GFP, new overview, and in live mode we will see the ER, but this time we are quite a bit now above the leaf, and we're looking at one of these outgrowths. Here the ER is very dense, and we will see the benefit of matrix detection. So let's see, we go to the scan menu, we make the pinhole a little bit smaller, just to keep it fair in comparison. We will also make the pixels smaller for super resolution. We will crank up the dwell time a little bit, and then we will activate STED. Or increase the excitation a little bit more STED power maybe. There we go. And if we scan this, we can see on the left the confocal and on the right the STED image. And while we can see in the STED image increased resolution, there's still a strong haze. So I'll switch to matrix, and as before I open the pinhole. So pinhole open, matrix on, scan, and when we are scanning, we will see again the virtual pinhole image. And I think what's striking here is that the background haze is really non-existent in the STED image. I'll scan again because I think things moved a little bit, and before we had more strings here. I don't know, scanning maybe another time just to see the difference. Going to post-processing, we can see with differential detection even less haze, and I think the STED image is a lot sharper than compared here to the confocal image and here matrix detection. So I think especially in STED mode we can see the difference here between matrix and confocal detection. Scan again, and I think now we're getting a few more strings here. Yes, that looks nice. There we go. Yes. So I'll stop here and scan once more. There. Now go to post-processing, differential detection, apply, and I think we can see and appreciate the reduced haze. Repair. I will repeat this exactly without matrix detection in a small pinhole. And in that case, we should be getting, again, much more haze. And you can see this is the case exactly here. So I think, just as promised, we can combine matrix detection and STED, and it removes a lot of the haze. And that's really the case because in STED microscopy, we are removing a lot of the in-focus light because we're switching off fluorophores, and we leave only the ones in the very center of the scanning spot to get the super resolution effect. So effectively, we have fewer fluorophores. However, what we're not doing with STED is switching off the autofocus light. And this is why matrix can be so helpful here to reduce the autofocus light and to still highlight the very highly resolved proper signal. I hope you enjoyed these examples of plant imaging, and I hope I could convince you that we have a few valuable tools for plant imaging in confocal mode using time gating and also spectral detection to really suppress your autofluorescence, and also in the super resolution area where we used matrix and STED and the combination of the two to do super resolution imaging, not only in fixed samples, but also in living plant tissue. And with this, I would like to hand over to my colleague Julia, and I'm happy to answer questions.
Thank you, Gero, for your talk and for sharing with us your super resolution images of plants. I believe that this technique will push the field of plant microscopy forward. And during your talks, a lot of questions came in from our audience, and let's start with the Q&A session. The first question is for Kathryn. Kathryn, do you have a preferred type of microscope for imaging plant samples?
Thank you for the question. We always use upright microscopes. We find it's very difficult to mount tissue on microscope slides under cover slips to work on inverted systems. And quite often in an inverted system we just cannot image deeply into the tissue. Therefore we use upright microscopes, quite often fitted with water dipping lenses. So it gives us the opportunity to have the lens, water and then the tissue and miss out the cover slip. So again, avoiding some of the diffraction problems that may be encountered.
So upright microscopes are your preferred one. And regarding this topic, I have a related question for Gero. Gero, you used an inverted microscope for your demonstration right now. And how did you manage that?
Well, I improvised and manufactured a little chamber, which is here, one of the samples. And basically I'm using a normal slide, a normal cover slip, and then I cut a few spaces and also sponges to keep everything together. These sponges have a little slit, they keep it together. They don't squish the leaf down too far to stress it, but yeah, this works. And I had good results with leaves. I can't really speak for other parts of the plants, but for the flat part of a leaf, this worked fine on this inverted setup.
Here I have another question for Kathryn. Kathryn, do you have recommendations for mounting plant leaves on the slide?
Thank you. Another really good question. We use really technical ways of mounting our leaves. We use double-sided sellotape to stick the leaf to the microscope slide. We find this is a really useful way of doing it. If you get the right sort of tape and make sure you don't get water floating the leaf off the tape, it will hold the leaf nice and still for imaging. So really technical, double-sided sellotape.
Okay, thank you for your answer. And we got from our audience one question related to Kathryn. Kathryn, does air in the leaf has an impact on imaging?
Yes, that's another challenging question. Yes, air does cause trouble within the leaf. It'll depend on the leaf type that you're using. Some leaf types have a lot more air spaces. And if you don't do something about removing the air and to help it, what will happen is as you're exciting your fluorescent proteins, as you're exciting your leaf, the air will expand and escape through the stomata, causing the leaf to move. So the way we usually deal with it, in leaves that will allow it, we will infiltrate the leaf with water. That accompanied with water dipping lenses usually allows us to have leaves that stay still during imaging. Another way we have used, if you've got leaf tissue that's intact, we have used things like perfluorodecolin that infiltrates the leaf. But as I mentioned in the talk, you can't use this with pathogens that release water because the perfluorodecolin is water immiscible, and then you start getting diffraction problems.
Here I have another question for Kathryn. Kathryn, which depth range you required for imaging your plant samples, especially together with your pathogen?
That is an interesting question that depends quite a lot on which pathogens we're looking at. For something like Phytophthora, the basic interactions can be going on between the plant and the pathogen at least 50 microns into the leaf. Some of the imaging I've been doing with bacteria infiltrated into the leaf, I'm imaging down at over 150 microns into the leaf. So again, it will depend on the pathogen. Some of the fungi, you're going down 1 or 2 cell layers if the fungus is filling the substomatal cavity. It will depend on the pathogen, so fairly deep compared with what is usually done with animal tissues.
I understand it depends on the pathogen and also, of course, on your sample. And related to this, I have another question for Gero. Gero, you used a completely different microscope. What was your depth range you reached during your demonstration, and what are the limitations?
I reached about 50 micron in these images which I was doing here on the machine. I was using a high-NA water objective, so this really helps to get deep into the sample, at least when you have a microscope which is nicely adjusted here, like this one. And I would say I could probably get even deeper, especially if I use a deformable mirror, which is one of our adaptive optics tools. So with adaptive optics, I believe you can get even deeper into leaves, unless they're completely opaque. And I think I've demonstrated it with 3D STED, which is traditionally the hardest part to get deep into the leaf. With normal confocal mode, I expect even deeper imaging really here.
And here I have one question for Kathryn. Kathryn, what do you expect from super resolution imaging in plants?
We haven't had the opportunity to do very much super resolution imaging, one of the major challenges has been the depth penetration. So being able to image deep into the tissue. The other challenge is how the systems deal with the autofluorescence from chlorophyll. Those are the two major challenges so far. We're looking forward to be able to do a lot more super resolution imaging.
Thank you for your answer. So I hope that you have more chances to do super resolution imaging with your samples. And here I have another question for Kathryn. Kathryn, how do you typically distinguish between autofluorescence and your signal?
I think wherever possible, we will use either filters or spectral detection to separate the fluorescent signal from the fluorescent proteins, compared with an autofluorescence signal, for example, from chlorophyll. We try and do it at the detection stage rather than post-acquisition. From my experience of trying to do spectral unmixing post-acquisition, it's rather challenging that a lot of the autofluorescence within plants can make it very difficult to have accurate spectral unmixing. So wherever possible, we will collect the images, separating the fluorescence as we collect them.
And we got here one question again for Kathryn. Kathryn, from your experience, how long can you image live plant tissue?
Yes, that's an interesting one. From experience, we've had colleagues who look at virus movement and have been able to follow the expansion of viral lesions even overnight. The main problem with plant tissue is that we can't do the same things as in animal tissue, as in we can't use the same types of autofocus systems. So in having to do long-term imaging, we have to work out a way of keeping the same plane of focus. That's the main challenge with the long-term imaging in plants.
And I have another question for Gero. Gero, regarding phototoxicity, is it possible to protect chloroplasts with adaptive illumination?
Well, I would say adaptive illumination does protect all structures of the leaf or of your sample, because we're using the STED laser only exactly where the STED laser needs to be active, meaning the STED laser is only on when there's an interesting fluorescent structure present. In all the other areas it's off. With the chloroplasts, obviously if they're fluorescing or giving an interesting signal, the STED laser will be on. If not, if there's no labeled structure in there, you will have the STED laser off and they will be protected. I have done something similar but more advanced in a paper, I think 2 years ago, where I did the reverse, really. I was looking at a signal from sensitive structures. In that case, it was malaria parasites, which also have sensitive structures. And I was specifically switching the STED laser off, where I got a signal from these sensitive structures. I think this could be easily repeated with plants because these chloroplasts are highly fluorescent. So if I hit a chloroplast fluorescent structure, I just switch the STED laser off and protect them this way. Yes, I think this is possible.
Gero, we got a couple of questions regarding the matrix detector. And one is about, does matrix detection support fluorescent time gating?
Yeah, it completely supports time gating. It's not a problem, so we can do the same autofluorescence suppression there.
Thank you for your answer. So definitely it works. And what is about spectral detection?
Yes, possible. I mean, we have been doing this right through this demo here.
All right, with this we are at the end of our Q&A session. We got a lot of questions and we were not able to answer all of them here, so we will come back to you via email. And if you got interested in our microscopes, or if you want to see your own samples on our microscopes, just let us know. And with this, we are at the end of our session today. Don't miss our next session about our live dyes and how to use them in live STED microscopy. And thank you for joining, stay healthy, and see you soon.